Neoplasia. A neoplasm is abnormal mass of tissue, the growth of which exceeds and is uncoordinated with the growth of the normal tissues and persists in the same excessive manner after cessation of stimuli which evoked the change. Neoplasia literally means new growth. Fundamental to the origin of all neoplasms is a loss of responsiveness to normal growth controls. In a common medical practice, a neoplasm is often referred to as a tumor. A tumor is a neoplasm formed in result of a typical pathological, uncontrollable, and unlimited growth of tissue, not connected with the general structure of a damaged organ and its functions. Etiology of tumor. Now, let us see the role of heredity in tumor growth. You see that from 5 to 10% of all tumors are hereditary. There even exist the so-called cancer families, but the most part of tumors are acquired. They are the so-called sporadic tumors, and they are associated with the action of carcinogens. Here you see examples of hereditary predisposition to tumors. In the left column you see syndrome, then you see tumor types, and the third column gives a genetic defect in gene. This is an example of a hereditary autosomal dominant syndrome associated with the development of malignant tumors. It is retinoblastoma in children, which in 40% of cases is hereditary. Another example of autosomal transmission of tumor is familial adenomatous colon polyposis, associated with mutation in the APC suppressor gene. One more example is neurofibromatosis. Here we see neurofibromas on the skin, tumors of the spinal roots, auditory and optic nerves, pigmented spots, and bone deformation. An example of hereditary autosomal recessive syndromes with impaired DNA repair. It is pigmented xeroderma. The disease is associated with absence or low activity of enzymes that eliminate the damaging effect of ultraviolet radiation on skin cells. This leads to inactivation of DNA repair proteins, which in turn leads to accumulation of defective DNA molecules and finally to development of skin cancer. Another example is Bloom's syndrome, which results from damage to both alleles of the BLM gene, which is responsible for maintenance of chromosomal stability. Patients with this disease present with erythema on the face and hands, decreased immunity, growth retardation, and a tendency to develop malignant tumors. Acquired tumors, they develop under the action of carcinogens. And here you see the role of carcinogens in tumor development. A carcinogen is an agent that, due to its physical and chemical properties, may induce irreversible alteration or damage to parts of genetic apparatus responsible for homeostatic control over somatic cells. It is the definition of Experts Committee of WHO given in 1979. There are known several carcinogenic factors: physical, which make 10%, chemical, which make about 90% of all carcinogens, and biological, which make about 10%. Physical carcinogens include ionizing radiations, which are electromagnetic waves with the wavelengths 25 nanometers and less, X-rays, neutron radiation, gamma rays, protons, alpha and beta radiation. Excessive insolation, electromagnetic waves with 400 to 800 nanometer wavelengths, ultraviolet radiation with a wavelength 320 to 280 nanometers, high temperature, mechanical friction. Chemical carcinogens may be classified into directly acting carcinogens and procarcinogens that require metabolic activation. Directly acting carcinogens include alkylating agent, acetylating agents. Procarcinogens requiring metabolic activation include polycyclic and heterocyclic aromatic hydrocarbons, aromatic amines, amides, azodyes, natural plant and microbial products, and other substances. This picture shows arrangement of carcinogens in the order of decline of their carcinogenic activity. Please pay attention that the most significant way of entry of chemical substances into the body is alimentary way. Biological carcinogens. They cause in humans about 10% of all tumors. In 1910, Peyton Rose first inoculated spontaneous chicken sarcoma using a cell-free extract. In 1933, Shope identified the Shope papillomavirus, which infects rabbits. His discovery later helped other researchers link the papillomavirus to warts and cervical cancer. By the beginning of the 60s of the 20th century, a Soviet scientist, Zilber, finally formulated the viral genetic concept of the development of the tumor process, according to which an oncogenic virus entering the cell introduces its genetic material into the chromosome of the host cell, becoming its integral part, genome or battery of genes, and thereby inducing the transformation of a normal cell into the tumor. The main biological factor is oncovirus. There exist over 150 types of them. Oncoviruses, after interacting with membrane receptors, lose capsids and are introduced into the cell in the form of a nucleotide. Here they multiply by replicating nucleic acid. Then the capsid is synthesized and self-assembled into the virion. DNA oncoviruses. Viruses of the Popova group. Popova means the first letters of papilloma, polyoma, and vacuolizing virus. They can cause both benign tumors, such as tumors of skin, anus, genitals, and malignant tumors of nose, mouth, larynx, cervix, penis, rectum. Hepatinoviruses cause hepatocellular carcinoma. Cytomegalovirus and HIV cause Kaposi sarcoma, B lymphoma, etc. You see a picture of hepatitis B virus. Epstein-Barr virus causes Burkitt's lymphoma, B lymphoma, nasopharyngeal cancer. Here you see a picture of Burkitt's lymphoma before and after treatment with cyclophosphamide, according to D. Burkitt. RNA oncoviruses. They include oncornaviruses, and retroviruses causing malignant, such as sarcomatosis, T lymphololeukemia, cancer of cervical mucosa, and benign tumors such as condyloma of genitalia, papillomas of larynx, and other tumors. Viral RNA genes integrate into the cell genome not directly, but after formation of DNA copies, which integrate into the cell genome, express, and cause its transformation into a tumor cell. Human T lymphotropic virus type 1, HTLV1, is a retrovirus that infects human white blood cells. It can cause diseases of the nervous system or leukemia. Pathogenesis of tumors. The basis of carcinogenesis is non-lethal damages to genes. The primary targets for carcinogenesis are three classes of normal regulatory genes: growth-promoting proto-oncogenes, growth-inhibiting suppressor genes, anti-oncogenes, and regulators of apoptosis. Most important are disorders in proto-oncogenes. Carcinogenesis. Carcinogenesis is a complex, long, multi-stage process which is based on a stable, irreversible pathology of genome and leads to acquisition by a cell of a tumor program of activity, with final elimination of the cell from the population. Stages of carcinogenesis include blastomocytogenesis, which consists in initiation or transformation and promotion, and blastomogenesis, which is third stage, tumor progression. Let us look at development of tumor in a simplified form. The main event is non-lethal mutation of somatic cells. If mutation is not eliminated by mechanisms of DNA reparation and involves certain genes, there is the probability for development of tumor. Of importance is damage to the following genes: proto-oncogenes, genes regulators of apoptosis, and anti-oncogenes. Here is the molecular mechanism of initiation of tumor, and in particular the role of proto-oncogenes and oncogenes. A proto-oncogene is a normal gene found in the cell. There are many proto-oncogenes. Each one is responsible for making a protein involved in cell growth, division, and other processes in the cell. If an error or mutation occurs in a proto-oncogene, it can turn into a malfunctioning gene called an oncogene. In other words, oncogenes are mutated forms of proto-oncogenes. Most, but not all, oncogenes in the body arise from proto-oncogenes. When a mutation occurs in a proto-oncogene, it becomes permanently turned on, activated. The gene will then start to make too much of the proteins that code for cell growth. Cell growth occurs uncontrollably. This is one of the defining features of cancerous tumors. There are at least 3 different types of mutations that can cause a proto-oncogene to become an oncogene. First is point mutation. This mutation alters, inserts, or deletes only one or a few nucleotides in a gene sequence, in effect activating the proto-oncogene. Second is gene amplification. This mutation leads to extra copies of the gene. Third is chromosomal translocation. This is when the gene is relocated to a new chromosomal site that leads to higher expression. Cell starts to grow out of control. Uncontrollable cell growth leads to tumor. Oncogenes and oncoproteins. Oncogenes code for production of oncoproteins. Oncoproteins may be nuclear, membrane, and cytoplasmic. Nuclear DNA-binding oncoproteins are mitogens. They stimulate cell division. Oncoproteins are homologs of growth factors. Oncoproteins are growth factor receptors. Oncoproteins are cytoplasmic messengers of growth signals. And there also exist nuclear regulatory oncoproteins. Synthesis of oncoproteins deranges the following physiological processes in a cell. First, binding of growth factors with specific receptors on a cell membrane. Second, activation of membrane receptors in response to growth factors. Third, transmission of signals to the nucleus through cytosol by second messengers. Fourth, production and activation of nuclear regulatory factors that initiate transcription of DNA. And fifth, activation of cell division. Suppressor genes and their role in carcinogenesis. Activation of oncogenes alone is not sufficient for complete transformation of a normal cell into a neoplastic cell. Uncontrollable division is prevented by suppressor genes, anti-oncogenes. They control production of growth inhibitors. Anti-oncogenes are P53, P16, P21, and other genes. Defective P53 gene was found in the majority of human neoplasms: breast cancer, cervical cancer, ovarian carcinoma, carcinoma of lung. Mutations of P53 lead to instability of genome and the loss of apoptosis by tumor cells, which gives tumor clones an advantage over other cells. Suppressor gene APC, adenomatous polyposis coli, is often defective in adenomatosis polyposis and in cancer of the large intestine. The common final result of action of different carcinogens is a neoplastic transformation of cell characterized by stable alterations in the DNA and by derangement of the interactions between oncogenes and anti-oncogenes in cell genome, manifested by stimulation of expression of oncogenes and or depression of anti-oncogenes. Most of the proteins encoded by tumor suppressor genes act as negative regulators of cell proliferation. Here you see the examples of the main known tumor suppressive genes. Carcinogenesis. So, initiation is accompanied by irreversible alterations in the genetic apparatus of one progenitor cell, in result of which this cell acquires ability to unlimited independent division, but so far it is not yet dividing. Then second stage comes promotion. Between initiation and promotion, different time may pass, just to several years. Promoters are factors which do not cause mutation by themselves, but activate transformed progenitor cell, which begins to divide. In result, a clone of identical cells is formed, possessing the same characteristics as the previously mutated cell. This marks monoclonal stage. Genetic material of tumor cells is unstable. That is why, with the growth of tumor, secondary and even tertiary mutations occur in cells, which is manifested by appearance of cells with different properties. So, in result, the tumor loses its monoclonal character. Appearance of several subclones in the tumor makes it polyclonal. This marks development of the third stage, tumor progression. Summarizing the above, we urgently recommend that you should thoroughly study the given block diagram. By biological significance for an organism, two types of tumors are distinguished: benign and malignant. Here you see the table of comparison between benign and malignant tumors. This table is obligatory for studying. Kinds of tumor growth. Depending on the kind of tumor growth, there are distinguished fast-growing and slow-growing tumors, from several weeks or months to many months or years. Another kind of tumor growth is destructive growth and growth without destruction, depending on the presence or absence of damage. Besides, growth may be with or without metastasis. In the picture, you see example of tumor growth. You see exophytic tumor growth, when tumor grows into the organ cavity, and endophytic growth, when tumor grows into the wall of a hollow organ. Expansive growth. Intensively dividing, mature cells provide a slow growth of the tumor from itself, leading to pushing back the surrounding tissues. This kind of growth is typical for benign tumors and for some malignant tumors, kidney cancer, fibrosarcoma. Infiltrative or invasive growth. Rapidly dividing immature cells lead to rapid tumor growth, capable of metastasis. This kind of growth is typical for most malignant tumors. Types of tumor growth. On the left, you see expansive tumor growth, when tumor grows in a limited space and simply pushes aside the surrounding tissues. And to the right, you see invasive growth, when tumor invades or infiltrates the surrounding tissues. Basic characteristics of neoplastic process. A very important characteristic is independence, which means escape of tumor from control by systemic and local regulatory mechanisms, with the result of uncontrolled growth. Then, potential unlimitedness. In 1961, Hayflick and Moorhead found that cells of mammalians normally make a limited number of divisions. This phenomenon of limited cell divisions is called cell aging or Hayflick limit. Neoplastic cells lose the division limit, which makes their population immortal. Immortality phenomenon. Other characteristics are loss of connection with specific function of the organ, inadequacy or loss of adaptation, hereditary fixation, which means transmission from mother cell to daughter cells, disorders in differentiation of cell elements, and presence of atypism, cataplasia, anaplasia. Cataplasia is emergence of poorly differentiated or non-differentiated cells resembling embryonic cells. Cata means downward. Anaplasia literally means reverse development. It implies loss of differentiation of cells. Morphologic cataplasia may occur on tissue and cell levels and is characterized by derangement of normal interrelations between parenchyma and stroma, by quantitative and qualitative alterations in size, shape, and structure of tumor cells, by rejuvenation of cells with return of their structure to embryonic organization, by polymorphism of cells, increase in size, appearance of shape non-characteristic of the given tissue, by increase in the amount of multinuclear cells, by increase in the number of chromosomes, by alteration of number, size, shape, and structure of nuclei, ribosomes, mitochondria, Golgi apparatus, and finally by alteration in the structure of cellular and subcellular membranes. Another kind of cataplasia is metabolic cataplasia, which is characterized by the following metabolic changes. Evident metabolic acidosis due to accumulation of lactate and other underoxidized products of metabolism in result of activation of glycolysis. Increase in the content of water, potassium, sodium, ferrum, zinc, and other ions, and of free radicals and peroxides. Increase in electric conductance of tumor and of negative charge of its cell. Depletion of tumor of calcium and selenium ions. Reduction in the viscosity of colloids in neoplastic tissue. Quantitative and qualitative alteration of synthesis of proteins in neoplastic tissues. Sharp increase in consumption of glucose by tumor and also of vitamins, lipoproteins, biologically active substances, and other metabolites. Elimination of inhibition of glycolytic oxidation of glucose in aerobic conditions. Negative effect of Pasteur. Reduction in synthesis and action of chalanes, inhibitors of stimulators and of non-specific inhibitors of proliferation, intracellular repressors, and other proliferation controllers, promoters. Increased synthesis and effect of different stimulators of proliferation, inductors, growth factors, inhibitors of inhibitors, etc. Immunologic, antigenic, and functional cataplasia. Immunologic or antigenic cataplasia is characterized by alteration of antigenic composition of tumor cells. There are several tumor antigens. Those induced by different carcinogens, they are strictly individual, polymorphous, and depend on the type of carcinogen. Embryonic antigens, proteins, for example alpha-fetoprotein, produced in result of antigenic reversion. Appearance of earlier lost antigens on the surface of cells. Antigens of viral tumors. If they are induced by one type of virus, they are similar for several tumors in different individuals. Tumor-associated transplantation antigens with characteristics of the antigens of transplanted organs that are produced in tumor cells in result of transcription of genes. Heteroorganic antigens. Antigenic divergence is acquisition by tumor of antigens inherent to other organs. For example, appearance in hepatoma of antigens that normally exist in kidney, and in tumor of kidney, adenocarcinoma, of antigens typical of liver and lungs. Functional cataplasia is characterized by alteration of specific functions of cell tissue structures. Malignant tumor has an ability to form metastases. or to metastatic spread. You see a picture which illustrates spread of tumor by hematogenic way. You see primary tumor from which some tumor cells detach, invade the environment, and penetrate the vessel by intravasation process. In the vessel, these tumor cells circulate and are delivered to some other place in the body, where they leave the vessel by extravasation and colonize this place, giving rise to a new tumor in this place, called metastasis. Factors of tumor resistance. Mechanisms of resistance to tumor can be divided into the following groups: anti-carcinogenic mechanisms acting at the stage of interaction of carcinogens with cells, organelles, molecules; anti-transformation mechanisms preventing transformation of a normal cell into a tumor cell; anticellular mechanisms committed to prevent formation of a colony of tumor cells. Here you see immunogenic anticellular mechanisms. Paraneoplastic syndrome is systemic disorders in an organism which do not depend on the localization of the tumor. They include cancerous cachexia, disorders in blood system, anemia, leukocytosis, thrombocytopenia, disorders in hemostasis, disorders in microcirculatory system, disorders in the structure and function of skin, mucosa, and other histohematic barriers, endocrine disorders, central and peripheral endocrinopathies, neurologic disorders, somatic and autonomic, central and peripheral neuropathies, etc. Tumor markers are predominantly normal differentiation antigens which many times increase in number in case of growth of tumor. These substances are used for diagnostic purposes. For example, marker of prostate adenocarcinoma is activity of acidic phosphatase of prostatic type, and one more marker is expression of prostate-specific antigen. Marker of hepatocellular carcinoma is alpha-fetoprotein. Marker of cancer of GIT is carcinoembryonic antigen. And marker of cells of mesenchymal genesis is in sarcoma is vimentin. Principles of treatment of tumor diseases. Surgical method. It consists in elimination of tumor within the healthy tissues using measures to minimize the scattering of tumor cells in the wound and to neutralize them. Chemotherapy, based on high sensitivity of actively dividing cells to damaging factors, and radiation therapy. Thank you for your attention.